This video is about light-dependent reactions, which is the first step in photosynthesis. But first, let's talk about light energy. The light energy we see from the sun is known as visible light. This is also the type of light that plants use through the process of photosynthesis to create chemical energy. Now, the energy from the sun is measured in wavelengths. You measure wavelength by the distance between two consecutive points in a series of waves. Now, different wavelengths have different energies. Smaller wavelengths have higher energies, while larger wavelengths have lower energies. Now, these different energied wavelengths also correspond to different colors in the visible light portion of the electromagnetic spectrum. As you can see, blues and purples have higher energy, while oranges and reds have lower energy. This light energy is utilized in the process of photosynthesis when pigments in plants absorb the light. Pigment molecules in plants can only absorb certain wavelengths and will reflect the color of wavelengths they cannot absorb. Chlorophyll A and chlorophyll B are two pigments found in plants that absorb light. More specifically, they absorb violet, blue, and red light, while they reflect green light, which is why many of the plants that we have are green. Another pigment that you would find in a plant is a carotenoid, more specifically beta-carotene, which absorbs violet, blue, and green light and reflects yellow-orange light. Photosynthetic organisms have a mix of these three pigments, which allows the plants to absorb energy from a wider range of visible light wavelengths. It basically allows for adaptation, which is why a cactus in the sun can thrive the same way as a plant can under the shade of a tree with only partial light. Now, how does light-dependent reactions work? The purpose is to convert solar energy into chemical energy, and more specifically, NADPH and ATP. To start it off, the light-dependent reaction begins in the thylakoid membrane, which is found in the thylakoid in a stack of granum. And you find that in your chloroplast. Now, there are two types of photosystems in the thylakoid membrane. You find photosystem 2 and photosystem 1, which are named for order of discovery, not order of function. As you can see, both systems have the same basic structure. They both have pigment molecules, a reaction center, a light harvesting complex, primary electron acceptor, and a chlorophyll A pair. Photons travel to the chlorophyll molecule in the reaction center. This chlorophyll donates an electron, which creates an excited electron. The energy transferred allows the electron to break free from the chloroplast and transfers to a nearby primary electron acceptor. The molecule of H2O splits to release a replacement electron in the thylakoid space. Now, this replacement electron allows chlorophyll to respond to another photon. As electrons move from photosystem 2 to 1, they lose energy. This energy is used to move hydrogen ions from stromas into the thylakoid lumen. Because of this, the electron must be re-energized in photosystem 1. Here, the electron is oxidized by another photon, which sends a high-energy electron to NADP+ to form NADPH. And we have our two energy carriers, ATP and NADPH. ATP is the buildup. It results from the buildup of hydrogen ions in the thylakoid space, which forms an electrochemical gradient due to uneven concentration of protons, and also causes different charges across the membrane. Potential energy is harvested and stored as chemical energy in ATP through chemoosmosis, which is the movement of hydrogen ions down electrochemical gradients through the transmembrane enzyme ATP synthase, which you can see right here. Hydrogen ions can pass through the thylakoid through an embedded protein complex, which is the ATP synthase. This protein creates ATP from ADP in the mitochondrion. Energy from the hydrogen ion stream allows ATP synthase to attach third phosphate which forms an ATP molecule through the process called photophosphorylation. The flow of hydrogen ions through ATP synthase is called chemoosmosis, which is when ions move from an area of high to low concentration through a semi-permeable structure. When looking at NADPH, which is the remaining function of the light-dependent reaction, we see an electron from the electron transport chain arrives at photosystem 1 and is re-energized with another photon captured by the chlorophyll, which was mentioned earlier. This energy from the electron drives the formation of NADPH to NAD, from NADP plus to NADPH. With solar energy being stored in energy carriers, it can be used to create a sugar molecule.